Hello, Peter Harty here from ARPANSA, presenting on some considerations for choosing a new primary standard to replace the current graphite calorimeter at ARPANSA. This calorimeter has been Australia's primary standard for absorbed dose to water for a number of years. In particular, in this talk, I will discuss whether we should continue with graphite as our calorimeter or switch to a water-based calorimetry system. I have no conflicts of interest to disclose. To start with, what is a primary standard? In metrology, it is a standard that is sufficiently accurate without needing calibration by other standards and is not subordinate to other standards. They're defined by other fundamental quantities such as length, mass, time, and temperature. They are also used to calibrate other standards, which are then distributed to other places. So why is accurate dosimetry important for us? The answer can be seen by looking at this graph of tumor control probability versus dose and normal tissue complication probability versus dose, with the line of ideal dose drawn as a vertical line, delivering high damage to tumor cells but low damage to normal tissue. Deviations away from this line by more than 5% are considered radiation accidents. ARPANSA aims to deliver calibrations to the radiotherapy centres at the 1% accuracy level. This means that when all the other sources of uncertainty are considered, the overall deviation is well under 5%. ACDS at ARPANSA has a role in this as well, doing on-site audits to check that the dose delivered by the clinic is acceptably accurate from both a dose magnitude perspective and dose delivery to the tumour site, taking into account all local sources of uncertainty. To ensure accurate dosimetry, there needs to be a primary standard which patient treatments are traceable to. Primary standards at ARPANSA are maintained under an authorization from the National Measurement Institute under the National Measurement Act. As part of this, ARPANSA participates in international comparisons and international metrology organisations and disseminates the standard to radiotherapy centres in Australia. For this purpose, the Australian primary standard calorimeter is maintained at ARPANSA, as pictured here. This calorimeter is placed into our Linac beams, as shown here on the patient table, and also in our cobalt tau therapy beam, to accurately determine the radiation doses for calibrating reference ionization chambers. The ionization chambers are used as secondary standards with traceability to the primary standard. The calibration process for direct calibration takes place like this. First there is calorimetry, followed by Monte Carlo conversion to absorbed dose to water. Then reference ionization chambers are calibrated in a water phantom to determine their calibration factors, which are then used to calibrate client chambers, transferring the calibration of the reference chambers to the client chambers. Later, when the radiotherapy centre wants to know their radiation dose rate, they place their ionization chamber in a similar water tank within their clinic. Using the same reference conditions, beam energy, depth in water and time, they measure the charge collected by the chamber. Applying the calibration factor supplied by ARPANSA for their very own chamber, they can determine the dose. Why do we need a new primary standard for measuring absorbed dose to water? The answer is that the present one is only good for photons, and we need the capability to measure high energy proton beams, which will be important when the radiotherapy centre in Adelaide opens. It's also important to future-proof the primary standard against possible failure of aging parts. The new calorimeter must be one that will have the following features: modern and suitable for the future. It needs to be usable for other types of beams such as protons. We're currently weighing up the relative merits of water and graphite calorimeters, both of which are possibilities. We have experience at ARPANSA in graphite calorimeters, but there are interesting features of water calorimeters which should be considered before we make our decision on how to proceed. Once the best way forward has been identified, PSDL will fully implement the new primary standard calorimeter over a period of 2 years. In general, water calorimeters are bulkier, heavier, and less portable than graphite calorimeters. This is because the calorimeter glass cells sit inside a large water phantom enclosure with significant mass from the water present. Total mass is of the order 80 kilograms. One possibility for water calorimetry is to build two water phantoms, leaving one at ARPANSA and the other to be kept at the proton therapy facility. The calorimeter glass cells could then be transported easily at the time of measurements. There are advantages and disadvantages which should be considered in looking at the features of graphite and water calorimeters. The major disadvantage of graphite is the fact that the medium is not water, and therefore there needs to be conversion to absorb dose to water. This is normally achieved through a Monte Carlo correction, which adds uncertainty. Water calorimeters don't need such a conversion. On the other hand, graphite has negligible heat defect, whereas this is something that needs managing for water so as to minimize the effect and keep the uncertainty low. Graphite has 6 times as much signal to noise ratio compared to water due to the relative specific heat capacities. Another difference is thermal diffusivity, which is 600 times higher for graphite than water, which leads to the measurement position being an average over the calorimeter core for graphite. But in the case of water, measurement is effectively at the points where the probes sit. And they must be in the beam, otherwise the absorbed dose is not properly measured. This leads to the question of what effects might be observable for thermistors sitting in the beam, which is a necessary part of doing water calorimetry due to the low thermal diffusivity of water. We have studied this using a graphite calorimeter designed for electron beams, shown here schematically with a thermistor sitting at the perimeter of the core. We took this calorimeter to the Australian Synchrotron for some measurements in their high-intensity X-ray beams. At the Australian Synchrotron, the theoretical spectrum of photons looks like this for one of their standard filtration sets. The energy distribution is lower than for cobalt or LINAC beams and has an average energy of about 80 kV. The high dose rates give high signal-to-noise ratios, making it a good place to study some beam effects. A typical calorimeter run is shown here for irradiation in the synchrotron beam, which has interesting steps at the beginning and end of the run, which are caused by scattered beam radiation reaching the thermistor. The closer the beam is to the thermistor, the bigger the steps. We have found that these steps do not affect the measured temperature rise, as the fits to the initial drift and final drift, shown here as dotted lines, completely determine the temperature rise, shown here as a red vertical line. This has been confirmed in numerous tests at various distances between the beam and thermistor. We have also looked for these steps in cobalt-60 and LINAC beams, which are at much higher energy, but haven't observed any steps. If they are present, they are too small to be observed. At the synchrotron, broad beams are created by moving one's detector either upwards or downwards in what is called scanning. In this way, a rectangular beam from the synchrotron, which typically has only 1 millimeter width, can be converted into a square beam with typical dimensions of 10 millimeters by 10 millimeters. Scanning upwards and downwards give very different steps in the beginning and end of runs. This will be seen in the next slide. Here are 2 synchrotron runs which we collected recently. The first has the beam close to the thermistor at the start of the scan and far from the thermistor at the end, while the second is the exact opposite, with the beam far from the thermistor at the start and close to the thermistor at the end. Although the shapes of the runs look completely different, the analysis shows that the temperature rise is the same for the 2 runs, as we can see by the sizes of the blue vertical lines shown. This is consistent with the hypothesis that the fitted drift before and after the runs completely determines the temperature rise. This gives confidence that the steps at the start and end of runs do not influence the fits to determine the temperature rise. In conclusion, ARPANSA is a choice, graphite or water calorimeter for a new primary standard. A new one, however, is necessary for us to have the capability to measure absorbed dose to water from proton beams. Each type, as we have seen, has its own advantages and disadvantages. Graphite, on the one hand, has high signal-to-noise ratio, negligible heat defect, is relatively portable, but requires conversion to absorb dose to water. Water, on the other hand, requires no conversion to absorb dose to water, but it has a lower signal-to-noise ratio, does have a heat defect, and is bulky and less portable. Since the thermistors in the new system will need to be in the irradiating beam, we did some studies of thermistor effects in beams. Observable effects in thermistor response were seen at energies such as 80 kV, but these sort of steps weren't observed at cobalt and LINAC energies. Steps in the thermistor response at the beginning and end of irradiation from scattered radiation do not change the measured dose rate, as long as correct analysis of the data is performed. Thanks for viewing my talk, and I'll try to answer any of your questions.